I'm really happy to introduce you to our two guest speakers this evening. We have Erin Arzola-Davis with us, who's a student at the University of Washington Environmental Science and Resource Management Program, and she'll be graduating in December. And the work she's presenting this evening was done as part of her undergraduate capstone. And I've also been very fortunate to work with Erin myself down here in the South Sound. She was a summer intern with Terrence Lee through the Nisqually Reach Nature Center. And so she helped coordinate pig use surveys on Anderson Island for 2 seasons. So I'm really excited to see her here this evening. And Dr. Sarah Converse has been mentoring Erin on her capstone and is also a longtime champion of our program and of the guillemots, completing research on Salish Sea guillemots using both our data and also her own through her own research projects. And she has also spent much time mentoring us and helping us dial in our protocols. Sarah is a professor at the University of Washington, and she leads the Quantitative Conservation Lab, where she works with students to develop models to better understand wildlife population dynamics and inform and improve their management. And as part of her work, she's been conducting research on pigeon guillemots on Protection Island. So she will talk to us a little bit about that this evening. So I will hand it over to you, Erin and Sarah.
Thank you, Shannon. And I can't start without just taking a moment to acknowledge Dave and Shannon and all they do for this project. They are absolutely both worth their weight in gold to this program. So it's always such a delight to see how amazingly professional you do with all this data. So thanks for that, and thanks for inviting me to be here. Dave really so nicely teed up what Erin is going to talk about, which is really exciting. So what I'll do is share a presentation. Hopefully this looks okay to folks.
Looks great.
Great. So yeah, we thought we would sort of do this tag team style. So we really kind of want to talk about a little bit of a mix of projects where sort of underneath all of them, we're really just trying to understand the relationship between pigeon guillemots and their habitat, which is, as we all know, changing. And so we'll talk a little bit about that today. And so kind of big picture, we're going to talk about sort of what we're trying to do with pigeon guillemots in the Salish Sea. Erin's going to talk about her work. And then I'm going to talk a little bit about Protection Island. I've talked to this group, I think, before about Protection Island, but maybe it's been a few years. So I'll provide some more updated info. And then I'll just finish up a little bit with some of the work to come. So just sort of big picture, I just wanted to talk about what we're trying to learn and why. So the first thing that you all know are that pigeon guillemots have been identified as an indicator species for Puget Sound. So the Puget Sound Partnership has identified them as an indicator species, what they call a vital sign. And because of that, it's really key that we really understand their relationship with their habitat so that we can understand the signals that we're getting from monitoring. What are pigeon guillemots telling us about what's happening in the marine environment and in the terrestrial environment in Puget Sound and in the larger Salish Sea? Another really important point about pigeon guillemots is, I mean, one, we're, I think, just so amazingly fortunate here that we have several alcids that breed near us here. That's not something that lots of people have access to, but we have several alcids that breed right here in the Salish Sea, which is cool. And some of those species are not doing as well as pigeon guillemots. We know that in general, the alcids are sensitive to a variety of threats, including climate change and human disturbance. So better understanding these threats will help us to conserve pigeon guillemots. And it may tell us some things that would help us to conserve other alcids in this region. And then last, and perhaps most important, is they're just fascinating birds. And better understanding their ecology enriches our lives and our understanding of this amazing place that we all call home. So those are kind of our motivations for this work. And with that, I'm gonna step back a minute and let Erin tell you about what she's been doing for her senior thesis. And Erin, you just let me know when you want me to forward your slides.
Awesome, will do. Can you hear me okay?
You can.
Okay, perfect. Thank you so much, Shannon and David and Dr. Converse for all that you're doing this presentation and celebration. This is my first celebration I think that I've been to. So yeah, super excited to be here. All right. If you want to go to the next slide. So Shannon already introduced me, but my name is Erin Arzola-Davis, and I'm an undergraduate studying environmental science and resource management with a focus on wildlife conservation at the University of Washington, doing a minor in marine biology. And I was a summer intern. I'm originally from Anderson Island, so I interned with the Nisqually Reach during the summers of 2023 and 2024, which is actually not where I was first introduced to the SSGN program. I was first introduced as a kid at summer camp that the Reach did that summer. So yeah, I've been watching the guillemots on the beach for many, many years, whether or not I had a data sheet in my hand. And so yeah, I had to do this capstone project as part of my graduation requirement in the School of Environmental and Forest Sciences. And I was super excited to learn that Dr. Converse was at UW and that she's involved in this program, especially because of how much data there is. Not a lot of people at a lot of seniors in my major get to work with such a like heavy data set. So that was an awesome opportunity. Just I'm also really happy that I was able to be involved in the collection of this data. So it really just brings this whole full circle moment where I'm able to produce something tangible from those many early mornings that I spent on the beach. And I'm ready for the next slide. So just to kind of reiterate what we already know about the birds and kind of set the stage for the research questions that I wanted to do. I really wanted to focus on the phenology of the birds, which is the timing of their life history events, particularly reproduction is what we're looking at since they breed here in the Puget Sound. We know that that timing affects their fitness. You need to maximize the resources that they have when they have it, because reproduction is very energetically costly. And we also know that some birds exhibit plasticity in their phenology. We know that climate change is a big driver of this change in breeding phenology, especially in polar seas. We see there's a lot of publications about that. And like Sarah already mentioned, we know that the guillemots are an indicator species here in the Puget Sound. So what I want to do for my study is I wanted to describe spatial and temporal patterns of their phenology in the Puget Sound. And I wanted to identify the scale of the drivers to see if it was maybe local or global drivers. And I'm ready for the next slide. So you guys already know all about the beach protocol, so I won't go over that. But basically, I took the big data set from, I think it was 14 years of data, 14 year data series, and I identified the highest count from each colony and year combination, and I used that to kind of identify when peak breeding season is. We talk about that a lot when we're trying to get enough people on the beach. Oh, it's peak. Need more volunteers this week. So I quantified that and used that week of year number as my main response variable. And I made a bunch of models to examine the influence of latitude, year, and sea surface temperature on that week of the peak count to see how those drivers might be affecting the timing of the breeding. And I use the count data particularly because it has the least amount of noise, as we say, when we're doing statistics. It's more or less pretty clear cut when we're doing the counts on the beach. So that we determined that would be the most reliable data to work with. I'm ready for the next slide. From a statistical standpoint, we found my top model was one that included both latitude and the survey year as the main drivers of changes in phenology. So we found a positive effect of latitude, meaning that the higher we went up north, the peaks were later in the year over those many years. And then we also found a negative effect of year. So as time went on, the peaks occurred earlier in the year. And I'm ready for the next slide. And we can see this modeled in these graphs these are estimates from that top model. We see that for latitude, for every 1 degree north, peak breeding occurred an estimated, I have it right here, estimated 0.86 weeks later. So over that latitude and a half, that was over an entire week's difference in peak between South Sound and the northernmost regions. This is a small effect that we found, but we would expect it to be a small effect considering that the range is so small that we're looking at. And that was also kind of gratifying since the South Sound protocols actually have us starting a week earlier, I think, than a lot of the other regions. So I thought that was cool. We also see in the bottom graph that stronger negative relationship. My model predicted that peak breeding occurred an estimated quarter of a week earlier every year. So over 14 years, that was an estimated shift of about 3.5 weeks. And then over on the right on this bottom graph, where those dashed lines are, those were oh, I guess it's not on this graph, but we had some future predictions showing years 2025 to 2027, but it probably in actuality wouldn't be quite such a linear relationship. We'd probably see it end up like evening out at the bottom, more of an upside down curve rather than completely linear. That's what we might predict to see in the future. There's probably some limit to how earlier their phenology will shift, that is to say. And I'm ready for the next slide. Just to reiterate the main points concerning this short exploratory study, the Puget Sound pig ewes have experienced a shift in their phenology over time, a forward shift to occur earlier. They have exhibited a latitudinal gradient in their phenology, where the northernmost ones are breeding a little bit later than the southern pig ewes. And this study also adds to existing global evidence of the flexibility of the seabird's phenology, especially in mid-latitude regions. A lot of the publications focus on those polar regions in the north and south, and there's not a lot of publications concerning these middle of the road seabirds. And this also adds to existing global evidence that seabirds are adjusting their breeding to occur earlier in the year. So another, if you remember from my research question slide, one of the goals I wanted to do was look at the scale of these drivers, which is hard to do just in this short project that I did. But the apparent local driver, which we found to be latitude, I think it would be interesting to look at that in the larger context of the Pigu's range, specifically up in BC, or even looking on, the other side of the Pacific Ocean to see what's going on over there, if that gradient is present or not. And then it could be important in the future to examine those larger scale mechanisms that is causing the guillemots to return to their breeding habitats earlier over time. And I think that is the end of my presentation. Thank you.
Thanks, Erin. I can keep going, and then maybe we can have time at the end and talk about all of it, if that sounds good. Yeah,
good.
I'm glad you got to hear about this work. This was really fun, and it's really fun to be able to use this big data set. I'm going to now sort of shift gears a little bit and talk about a different place and a data set that exists at a much different, much smaller scale. And this is a work we've been doing at Protection Island. And this work has all sort of been focused on understanding threats to foraging habitat in the Salish Sea, as well as how those threats might be affecting the population. And this has been a pretty big project, lots of collaborators on this, lots of funders. So the folks here at UW who worked on this, Liam Pendleton, who was a former graduate student, Alex Conan, who was an undergraduate who was also a technician on the project, Eric Wagner, who's a research scientist in biology, and Matt Farr, who's a postdoc in my lab. So our big kind of collaborators on this, Scott Pearson from WDFW. Many of you have kind of come across Scott or heard from him on calls like this before. Sue Thomas, who is now retired, but formerly Fish and Wildlife Service Protection Island and the Washington Maritime Refuges biologist. Another retired biologist, Lee Robinson, who did the original work on Protection Island that I'll mention. And a lot of other people who have really contributed to this work. And also, I want to acknowledge a lot of organizations that provided funding: the CDOC Society, the Fish and Wildlife Service Pacific Birds Program, and the Coastal Program for Region 1, which is the Pacific region, the Science Support Program in USGS, the American Wildlife Conservation Foundation, Wilson Ornithological Society, and the School of Aquatic and Fisheries Sciences at the University of Washington, as well as in-kind support from WDFW, Fish and Wildlife Service, and the Washington Co-op Unit, which is the organization I head at University of Washington. Okay, so this is Protection Island. This is going to look pretty familiar to those of you who are in Jefferson or Clallam County. Basically, this Protection Island is in Jefferson County. This is the Quimper Peninsula. Port Townsend's right here. This is the Miller Peninsula. This is Clallam County, as is this. Clallam County and Sequim is here. Everyone, I think, probably recognizes Dungeness Spit. So just this little place right here. It's a protected island, and really has largely been protected because it is so important to seabirds. There are an estimated 1,200 odd guillemots that nest on the cliffs and in the shoreline debris, woody debris. This is a really critical island for rhinoceros auklets, largest nesting site in the Salish Sea, about 36,000 pairs estimated. There are large numbers of pelagic cormorants and Olympic gulls, and there are also small numbers of black oyster catchers, and unfortunately very small at this point numbers of tufted puffins. Really just less than a handful of tufted puffins that are now to be found at Protection Island. This is just a nice photo from the area. You can see we come kind of from the south usually. Sometimes we come from the east, and we typically come in here to this little marina right here. And as you come into this marina early on a summer morning, all you can hear is gull calls and guillemot whistles. And it is a really cool, cool place to be. Other sort of neat things about this spit over here is a pupping area for harbor seals. We also see elephant seals hauled out there pretty regularly. It's a pretty cool place. And then here's just another look that you can get at it from one angle, where you can actually see kind of the variety of habitat. This kind of habitat here, this is rhinoceros auklet habitat. This habitat over here, this is guillemot habitat. This is, or was at one time, puffin habitat. And then along the shore, you get this woody debris, and the guillemots also really like this habitat. And then just another view. So now here we're looking back towards Squim. And of course, as I mentioned, it's a protected area. Most of it is owned by the Fish and Wildlife Service. It's now actually managed by Jamestown Sklallam Tribe as of this year, which is really interesting, and they've already been good partners. So I'm excited to be able to potentially work with them more in the future. And then part of the island is also owned by the Washington Department of Fish and Wildlife. So we're really trying, at least today, I'm going to talk about two things that we've been trying to do. First is understand a relationship between foraging conditions and demography. I'm going to really, in particular, focus today on breeding success, although we're also working on survival. And identifying specific critical foraging sites used by pigeon guillemots at Protection Island, and determine the characteristics of that foraging habitat. So I'll talk about the first one first, really focusing on this question about how do oceanographic conditions, how do those marine conditions influence the breeding success of guillemots? And so just a couple of photos here. Obviously, we are banding the guillemots. So you can see here a combination of a metal, USGS metal band, as well as these colored bands that are in combination. So if we can recite them sitting on the beach, we can get recites without recapturing them. This is a guillemot egg. Many of you will know what this looks like. These are some of the nest boxes that we use. These are sort of portable nest boxes that we put out every year. There are also 13, or actually 12 now, permanent nest boxes that are actually dug into the ground in the marina. And then here we're doing some measurements on an adult. So we have these permanent nest boxes, and then we also put these temporary portable nest boxes out in the woody debris along the shoreline. And these birds take to these boxes pretty readily. And so once we put them out early in the season before the birds want to start setting up shop. And then we monitor them weekly from pre-laying through fledging. We also, I mentioned that one of our collaborators is Lee Robinson, a Fish and Wildlife Service retiree. She worked and did this kind of work actually for 18 years at Protection Island, which is amazing, and has given us access to her data set. And then she monitored 410 nests and banded 564 chicks during that time. And then we have since added 3 years, 2022, 2023, and 2024, where we've monitored 17, 23, and 23 nests, and we've banded 8 through 13 and 18 chicks in those years. So sort of getting into how do we link nesting success to outcomes. So typically what we would do is when we make that first nest check in early June, late May, we're going to see often something like this in a nest box. This is one of the boxes that's actually dug into the ground in the marina. You take the lid off of it, and there's a couple of Guillemot eggs. And often if there's a couple of Guillemot eggs, there's also an adult in the nest. At some point, those Guillemots hatch out, and we've got chicks in the box. The chicks go from nice and pretty and black and clean when they're young until just before fledging, when they are in need of a bath. So fledging is good for them. They get to go out and have a bath. And so then at some point we keep coming back and the box is empty. And so then what we do is we say if the minimum age of the birds is at least 29 days, we assume that those chicks fledged. And we record outcomes of 0, 1, or 2. And I'll tell you what these mean in a minute. So 0 is we expect that no birds fledged. 1 is 1 bird fledged, and 2 is 2 birds fledged from that nest. And then we model these with these, scientists can't help but use Greek symbols. So we model these with these probabilities. So B is the probability of producing at least 1 chick. So if we get no production, then we have that probability is 1 minus B, the probability of getting at least 1 chick. We can also get just 1 chick produced. So yes, we got 1 chick, so with probability phi, but we did not get a second chick, and we give that a probability psi, and so we did not get that, gamma, so we did not get that second chick. And then we also have this happy outcome where we got our first chick and we got our second chick. So you can think of this as, with people, right? This is the women who don't have any children. This is the women who have a single child. And this is the women who have multiple children, right? Multiple births. And so we're doing basically the same thing with our Guillemots. And then we're modeling each of these probabilities as a function of oceanographic conditions in a given year. And we looked at 4 different oceanographic indicators, 2 of which are local and 2 of which are large scale. So we looked at chlorophyll, which is a local predictor of productivity, as well as the North Pacific gyre oscillation, another predictor at a much larger scale of ocean productivity. Another local predictor is sea surface temperature, and the Pacific decadal oscillation. So these are the hypotheses we had about what we expected in terms of our breeding success and possibly our sort of twinning probability, the relationship that we expected to see with these marine indices. And so we, as Erin talked about, we fit these models and we assess sort of the strength of evidence for the models in the data. And we fit a lot of models, and what I'm showing you here are really the only 2 models that had sort of a meaningful amount of support. So we talk about the weight of the models within the whole data set. So these 2 models accounted for 40% of the weight, and then all the other models we ran accounted for 5% or less of the weight. So we think that these are the most supported models. And the big picture thing here is they're pretty simple models. So we put year in every model. We basically allow for this residual annual variation. And so that sort of kind of cleans up what's left after we bring in these covariates. And the only covariate we found support for was the North Pacific gyre oscillation. And we saw that this had some impact on the probability of nest success, the probability of producing at least 1 chick, although it wasn't hugely more supported than a model that had no effect. So in essence, what we found was that there is annual variation, but we couldn't really tie it strongly to any of these oceanographic indices. This is what sort of our estimates for these different probabilities looks like. So this is the probability of having at least 1 chick. So this is sort of equivalent of that sort of nest success that Dave talked a little bit about. And when we get in and we monitor them every week and we see what's going on, what we get at Protection Island is about 60% or so of the nests produced, that the nests that are defined by a place, a nest that had an egg or 1 or 2 eggs in it at some point. That's the probability that those nests produce at least one fledged chick, I should say. And then this is the probability that, given you fledged at least 1 chick, the probability that you fledged 2, and that's about 25%. So what we're seeing at Protection Island is, so for those nests that are successful and fledge a chick, about 75% fledge 1 chick and about 25% fledge 2, even though the vast majority of those nests start with 2 eggs. And this is what the relationship with NPGO is. So this is sort of high NPGO years, low NPGO years, and we see this relationship. And again, NPGO, the North Pacific Gyre Oscillation, is an indicator of ocean productivity. So we do see this relationship with ocean productivity. And as I said, we also see this strong variability from year to year, especially in this nest success variable, much less in this twinning variable. That's pretty constant at around that 25% from year to year. So it might be that this sort of nesting success is, maybe this is more of an individual variation thing. Some guillemots are just experienced enough and good enough to be able to pull off those 2 chicks. And the sort of just the ability to pull off any chicks is really more tied to what's going on in the environment. So, as I said, we found relatively little influence of these oceanographic indices on demography. A couple of points about that. We did look at a couple of Pacific-wide indices, and Protection Island, of course, being inside the Salish Sea, is somewhat sheltered from those. Although we do find in some other places, in some places like the Salish Sea, that those Pacific-wide processes can be important. And I think maybe in some ways more significantly, these guillemots are generalists. They really do, of all of our alcids here in Puget Sound, they do have a little bit more of a generalist capability and the sort of potentially the ability to switch between prey types. Although it is interesting seeing how constant they've been in their preference for gunnel and sculpin over time. Okay, so that was sort of the first topic, and then the second topic is this work that we're doing on foraging habitat selection. So this right here that you can see, this is the back of a bird, and this is about a 4-gram tag that collects GPS points. And when the bird returns to the island, it transmits those GPS points to a base station that we put in the area. And we tape it onto the bird. It doesn't last very long, and it falls off because it is just on there with waterproof tape. But these birds seem to do really well with these, and they go out and they tell us where they're going. And so the goal here, as we said, is to understand what that important foraging habitat is. So we put these tags on 13 different guillemots. This work is also, we're doing this similar work with rhinoceros auklets at Protection Island, but I'm just talking here about the guillemots. And I'm not going to talk so much about the analysis right now. What we're doing is we're trying to fit what are known as resource selection functions so we can understand what they seem to be selecting for. But I thought what I would do here is just show you some maps of guillemot movement. I thought that might be most interesting. So again, this is Dungeness Spit, the Miller Peninsula, the Quimper Peninsula, Protection Island. So I'll just give you for scale, if you're not super familiar, sort of the tip of Dungeness Spit to Protection Island is about just a little under 9 miles. And so here you can see a guillemot moving around. I've got the dates at the top. This is over about a 4-day period, 3-day period. this guillemot moving around, staying pretty local. This is a very common thing that we see these guillemots moving over to the Miller Peninsula, and I'm interested to hear what Jefferson and Clallam County folks think about this with respect to the places that they're surveying. And a lot of this sort of foraging energy, probably foraging energy right offshore here. Another bird that stayed pretty close to home. This is clearly an errant location, don't believe that they're going that far inland. Another bird that's going over here closer to Dungeness Spit. We saw this pretty commonly, similar kind of thing here, birds that are moving over towards that direction, and similar kind of thing here. These birds are probably sitting and drifting on the current, which is why you probably get these kinds of patterns. So yeah, we're working on analyzing these data, as I said, bidding. First of all, what we're trying to do is understanding what of these points represent foraging, and we do that by fitting movement models that work to distinguish different kinds of behaviors. And so what we expect when birds are foraging is that they're moving kind of in tight clusters of points. And so we're looking for that kind of behavior. Then once we identify that foraging behavior, we can say, what is it about the places that they're foraging versus places where they could have foraged? And so this is sort of still ongoing work, publications to come. We're very close to submitting the Protection Island Nest Analysis. This is being led by Leanne Pendleton, a former master's student, as well as a postdoc in my lab, Matt Farr. The movement analysis that I've just been talking about, those same two folks are working on that. Erin talked about her work on phenology, which we would like to turn into a publication. And then some of you will remember a former PhD student of mine, Amanda Warlick. She's now a scientist with NOAA here in Seattle, working on marine mammals. And Amanda was working on, and Dave made reference to this, using the burrow visit data to understand nest productivity. And that is a publication that's just about ready to submit to a journal. And then I just wanted to mention a couple of new things. In addition to the potential to continue at Protection Island, a couple of weeks ago, I had a really nice opportunity to visit Jack Island and Bendovi Island. Some of you will know that Jack Island is just north of Guillemot Island, and Bendovi Island is a bit north of there. And these two islands are owned by the San Juan Preservation Trust. And so been in conversations with Kathleen Foley-Lewis, who works for the trust. And then we got to go out and visit with Kathleen and Thyatira Thompson, who is the caretaker of Bendovi Island and has really an amazing job to get to stay at Bendovi Island. And they are working on trying to do some restoration of some shoreline areas at Bendovi. So we would like to understand maybe what's going on there. And Jack Island is a pretty nice guillemot nesting area. And so it offers up some opportunities to dig in and understand a little bit more of this ecology of guillemots in the area. I'll also mention that there's a lot more to do with the Salish Sea Guillemot Network data. I think we've only just scratched the surface. So yeah, thank you for letting me rattle on quite a bit. I'll stop and open it up to questions, and including especially questions for Erin. Thank you so much.
Thank you so much, Erin and Sarah. That was really wonderful. And I just love that you guys have been digging into our data, getting in there and finding ways to analyze it, you know, more than just summarizing numbers, but actually looking at what's happening with the birds. And Erin, really well done. I was amazed at how significant the shift is in time, in a relatively short amount of time and over a relatively small area. So that's pretty incredible. Good job. Do we have any questions for Erin and Sarah? Dave, I see your
Yeah,
hand.
yeah, I have a question and we've noticed at our colony that late in the season, we have juveniles that are fishing very close to the colony, just a little bit south and in the near shore. But the parents that are provisioning chicks seem to fly in from the west. And I've wondered why, why aren't they fishing the same young that the juveniles are? Why are they expending so much energy flying away?
Yeah. I so a couple of things I'll say. I mean, partly that's what we're trying to sort that out. Of course. Although what I will say is that one of the challenges of being a seabird ecologist is that the very thing that our study animals care most about is something that we can rarely see or get information on, right? So they are almost certainly what they care about is where are the fish? And so why are the fish consistently from year to year more likely to be seen off of the off of the mainland versus sort of right offshore from Protection Island? I don't think we totally know that, but we're certainly trying to dig into that and looking at things, looking at different predictors. We are also looking into potentially getting some data from forage fish surveys, but what we generally find is that the forage fish surveys are not as frequent or as regular as we would like them to be to really be able to dig into them and sort of hold them up against these long-term data sets. But yeah, it's probably some combination of depth and currents and other sources of nutrients that may be impacting where the forage fish are, and the birds are probably where the forage fish are. Yeah, it's a great question though. It's really hard to know, and it's yeah, seabird biologists all over the world are constantly looking at these indicators that we know are kind of not the thing that our birds actually care about, but because we don't get to see what birds actually care about, we're looking for sort of, things that might be correlated with them.
Thanks, Sarah.
Yeah.
Al, I see you've got your hand up.
We had observed at our beach that the species of fish delivered were almost burrow specific. I mean, in our area, it was almost all gunnels. If anybody brought in sculpins, it went to like a single burrow. So with these birds that you're tagging, are they coming back with a variety of fish, or are they targeting specific species more than others?
Yeah, that's a great question. I'm afraid we don't know that. I think probably, and I don't know that they've dug into this yet, although we are trying to find a way to work with folks in the Farallones who do have sort of long-term observations. And I think maybe you've even heard from Mike Johns on one of these calls, long-term observations of foraging. It would be really interesting to know. One of the things that fascinates me about animals is I think as people, we all recognize our individuality, but with the animals we study, we tend to say, oh, they do this or they do that. But there may be a lot of individual variation, and that individual variation may itself be a function of learning and experience, right? If I'm a guillemot and I'm trying to carry off a nest, and I had good luck one year foraging in one area, I may go back to that area over and over again. We don't really have the kind of data that can help us figure that out, but we certainly see that in other species. And over time, we may be able to get the kind of data that might help us figure that out. It requires us to have, multiple year continuous data from individuals so that we can see, do we really see individuals doing different things? And it would be very interesting to have long-term data for individuals, and see sort of how they build up their experience and their behaviors over time. That would be amazing, but isn't in our grasp yet with birds like this. But yeah, I think those are really interesting observations, and like I said, I'm very interested in the potential that those are sources of individual variation that may be learned. Yeah.
And Erin, Maria Ruth has a question for you. Can you confirm what I think you said, that the peak numbers of birds between 2012 and 2024 occurred 3.5 weeks earlier? And if so, do you think SSGN surveys should start earlier?
Yeah, Maria, thank you. They did occur, according to my model, about 3.5 weeks earlier. I think it was pretty close to what actually happened. If I'm remembering correctly, the peak in the earlier data, was happening around late July, early August, and in these most recent few years, peak has been occurring closer to the beginning of July. So I think maybe this surveys, I don't know if they're like arriving earlier. I know that every time for the 2 seasons that I surveyed, um, especially in 2023, there was already a lot of activity. So that might be an indication that we need to start the surveys earlier, but I don't want to make anyone groan at that advice.
Something to consider for sure. And I think
Yeah.
especially colonies who see that early activity, yeah, there's no reason why folks can't start earlier. So it's definitely something to think about.
Mhm.
Yeah, Francis, I see you've got your hand up as well.
Yeah, thanks. And Erin, thanks so much for that presentation. We've often found here on Whidbey Island that south-facing borough colonies tend to start a little earlier as well. I don't know if that's something to consider for the future. And Sarah, it's really great to have you here again, and thank you so much for that presentation. My question really is, as I was listening to Dave's results, thank you, Dave, and also what you were saying about Protection Island, Sarah, it seems that the there is a higher success rate with the fledging chicks there on Protection Island,
Hmm.
and the you have the opportunity of looking into the burrows and really figuring out what's going on there. So there's that going on. But is it your sense that that we're not catching that success rate to create it, to come up with it as high as yours is, or are there just too many other factors, disturbances, the fact that you're on an island without predators in the same sense that what we run into with our surveys?
Yeah, that's a great question. I was thinking about that when I was looking at Dave's numbers, and it is hard to disentangle these two things, right? Protection Island is a very different environment, but we're seeing success rates, in the 60+%. And so again, that's the probability of producing at least one chick. But I think that's probably pretty equivalent to what Dave was showing, and using the calculation method there. I think it was closer to, it's about 30 or something, wasn't it, Dave?
Yeah, so it was about, on average, about 25% at Whidbey, but around 30 at some other regions. Yes.
Yeah.
And we're
So,
not, I'm sorry, go ahead.
oh, yeah, I mean, I'll just say, like, I think it could be both, either or both, but I will share that, yeah, what we do see is that those visits become fewer and farther between. It's harder to catch the adults sometimes, in the nest as those chicks are getting a bit older. And so, it might be that it's just harder to catch those late deliveries. What we haven't been able to spend enough time at the island for like long periods of time during the day to understand the shift in that delivery. But again, we do have the opportunity maybe to dig into looking at some of what they are learning in the Farallones, where they're able to observe these birds for long periods of time and get foraging information. This also offers up opportunities for places like, as I mentioned, like Jack Island. We could dig into some of these things potentially. Putting cameras on nest boxes offers all kinds of opportunities where we can actually know when adults are visiting. So my guess is that, I don't know, my guess is that your nesting success isn't that much lower, because if it was, like it's hard for me to imagine that the birds would be as successful as they are there. And so that makes me think that it is probably, at least part of it is missing late and infrequent deliveries a little bit. And this was kind of one of the things that Amanda's trying to do with her modeling too, is sort of try to account for this and see if we can account for this sort of detection issue, which is such a big deal in wildlife studies. They don't want to be seen. We don't always see them. So we're trying to constantly sort of figure out how to account for that. Yeah, great question.
Yeah, thanks, Sarah.
Yeah.
It's tough. We can't just pop the lid off the top of the burrow
I
to see
know.
which burrows have eggs inside either. So
Exactly.
our active burrows, which we're using as sort of a measure of breeding activity, we're not sure if they've laid eggs in all of those burrows or if they're just visiting.
Yep.
So yeah, that could skew it just a little bit as well. So.
Yep.
All right, well, we've gone over time, but we really appreciate you staying on and answering a few questions. It's really fascinating information, and I'm happy that there's so much work being done with guillemots in the Salish Sea. And Erin, you mentioned Canada. It would be nice to add some of that data, and hopefully we'll be bringing BC into the survey. So within a few years, we could have some data coming out of the northern Salish Sea as well. So it would be interesting to sort of run that analysis again and see how it's different.
Yeah, that would be awesome.
Yeah, thank you both so much for being here this evening and for giving us these wonderful presentations. We really appreciate it.
Thank you, and thanks to all of you for all your work. It's really fun to be able to benefit and play around with all the data you've worked so hard to collect.
Agreed. Thank you, guys.
Good to see you, Erin.
Good to see you guys.
And with that, we've come to the end of our evening. Thank you, everyone, for taking this time this evening, spending it with us. We had a wonderful season. It's so great to see everybody's faces and just to come together all at once here to celebrate our birds. I hope you all had a wonderful season and also enjoyed your time out on the beach, connecting with not just the guillemots, but all of the other wildlife and even the eagles and the seals, and just connecting with the Salish Sea. There's our Camino group there. Glad you guys are able to join us. Hope you had fun tonight. All right, well, thank you, everyone. Have a good evening. Take care, and hopefully we'll see you next season.